Hi everyone, is my audio clear?
Yes, sir.
Yes, thank you. Good afternoon everyone. And in today's class, we will discuss about prostate cancer stem cells. And I'll quickly have a review on cancer that we have discussed about and about cancer stem cells. And then I'll discuss about prostate cancer a little bit. And then we'll try to learn a few things that are specific to prostate cancer stem cells. As a part of cancer stem cells, the third unit, we will discuss a few, 3 or 4 different cancers. There will be redundancy in terms of molecular mechanisms or probably the properties and so on. So try to focus on what is different or more for each of these cancers. Okay. So we already described what is cancer. It's neoplasia, uncontrolled cell growth, cell cycle dysregulation because of which cells grow more. And in general, if we look at cancer normal cells, if there is any kind of cell, and then the cells have a certain fate, and each of them, some of them will not be produced anymore, some will stop dividing, and they will go undergo apoptosis. But mutations may happen which will allow further production, or those are mutations in oncogenes and or mutations in tumor suppressor genes. I think I have described these, but again, oncogenes, because of a mutation, if they gain a gain of function mutations, that means they are hyperactive. Oncogenes will allow cell division. So oncogenes, if there is a mutation because of which cell division is enhanced, then the cells will become more and more. There is also another set of genes. Those are tumor suppressor genes, and they activate cell death, or at least they prevent cell division. Okay, we will put it as prevent cell division. So if there is a loss of function in these, if there is a mutation because of which there is a loss of function, then these will not be working, because of which cell division is not prevented, because of which the cell keeps on dividing. So these are some of the way this is one way of how you can look at cancer also at the level of genes and so on. Oncogenes and tumor suppressor genes. Then we also discussed about the nature of the tumors: benign, which are slow, non-invasive, and they're not metastatic, whereas the malignant ones are much more rapidly growing, invasive, and they cause serious damage because of metastasis. Tumors are heterogeneous, meaning that there are more than one cell type, right? Which means there could be different cell types within one tumor, and out of them, not all of them can cause cancers, and it's a small subset which are referred to as the cancer stem cells. And we also discussed about FACS and staining, Hoy's dye, and the site population we heard, right? And we also learned about CD133 and CD44. We also have seen a table where CD24, and there are integrins and many other as well, many other cell surface markers. But these CD133 and CD44 are most prevalent in majority of the cancers thus far characterized. They also have a property of spear cultures. They can form spears, similar to that of what we have learned about embryoid bodies. So there are 3 important properties of cancer stem cells. Those are tumorigenicity, self-renewal, and differentiation. These 2 are stem cell-like properties. You can think of it like this. These 2 are properties of stem cells. And this is the property of cancer. And that is how cancer stem cells. Okay. And why are we studying cancer stem cells is because we know that the tumors, majority of the times, they relapse. That means we treat with conventional therapies such as chemotherapy or radiotherapy, and then after some days or some years, a couple of years, the tumor will relapse, and then that keeps on growing and into metastasis and so on. We don't want that kind of things, or we want to find a cure so that the cancer does not relapse. In that case, we have to somehow be able to delete or kill these cancer stem cells, because the rest of the cells are by themselves, they cannot um, keep on growing, so they will all go, they will die eventually. So targeting cancer stem cells is will become, or is the central idea to eliminate cancers completely. There are problems, of course, but still that's the goal, at least. So now we are going to discuss about prostate cancer. We will get to the biology of it, and it's important that because it's general biology of humans, we should try to learn a lot more about it. So there are about 2 lakh deaths worldwide each year, and there is a loss of homeostasis, which is characteristics of any any cancer. And that is loss of homeostasis. I mean, the cell number will increase, and that is where the organ keeps on growing, the prostate gland. We will see it in a few slides, maybe in the next slide. So some of the things that are involved in that seems to be enhancing this or the incidence of prostate cancer happens to be with inflammation, diet, and some people are genetically predisposed to this cancer. So it is quite common. It's not very common, okay? It is, but as people grow, as men grow older, there is a larger probability. So prostate gland is somewhere here, if you can see it. That is the prostate gland, right? It is in between. You can think of, say, see the bladder. This is where the urine is collected, and this is the male reproductive organs, the testis, and this is where seminal vesicle, that is where the sperm is stored when it is present in more, and prostate gland is present here in the junction between where the sperm and the urine. This is the same way for outlet, and that is where it is located. It is a non-essential organ. What do you mean by non-essential? If you remove the prostate gland, the human can still survive. Okay, it's something like if a hand or finger is lost, still a man can survive. Not if kidney is removed, or lungs are removed, heart is removed, or brain. Those will become the essential ones for the human to function at all. So majority of the organs that are in our torso are all the essential ones, right? In the brain and the torso. So its function seems to be to keep the sperm viable by the production of some prostatic fluids and so on. It is thought that it will keep the viability for a longer time. The prostate gland has a problem, not a problem, but the androgens are produced, and that keeps the prostate gland active. Okay, prostate gland is one of the rarest of the tissues that has possibility of growth throughout the life. That means a boy with a small prostate may possibly have a larger prostate as they get older. Okay, so that is they grow very slow. Of course, it is there, and they grow very slow. And there are many other factors the way the prostate gland is influenced by these androgens. Androgens are something like the sexual hormones like testosterone, for example, which can induce the growth of prostate gland. So there is what we have to remember is there is a predilection for prostate gland to keep on growing, right? Keep that in mind. So if we look at in the closely at the prostate gland has several alveoli-like structure. They have secretory cells. These are the secretory cells which secrete the prostatic fluid into the lumen. That is the ductal lumen. That is the gap that is present. And the lumen consists of the prostatic fluid. So there are two types of cells. One is the outer, this basal cells, the layer of cells on the outside, and these are the cells that are the secretory cells. The function is to secrete the prostatic fluid into the lumen, right? One will find several of these ducts and several of these lobule like things which will all form into one channel, and then the prostatic fluid is collected in there and secreted along with the sperm. So what we are discussing is about, we said the androgens are important, or kind of they allow the growth of these the cells that are present in the prostate gland. So there are two compartments, as we discussed. There is basal cell, and there are these secretory cells here, right? So this is a schematic trying to show the basal compartment is here, and this is the luminal compartment. Those are the cells that where the luminal cells, the glands cells that secrete the prostatic gland are present. So the basal compartment apparently has the stem cells. I hope you also remember I was putting the stem cell and the design of general tissues. If you look at it from the perspective of the adult stem cells, is that they all have the stem cell, and the stem cell occasionally may divide, or whenever there is a need, and produce a cell, and that is transit amplifying cells. They proliferate rapidly, and eventually they give rise to terminally, differentiated cells, right? So the basal compartment apparently has the stem cells, which will now and then divide and give rise to differentiated cells like this. Okay. So what you want to remember here is the basal compartment, the stem cell, is usually androgen independent. That means for the survival of the cells in the basal compartment, or the stem cells at least, they do not need any androgens. The transit amplifying cells are kind of androgen responsive. That means if the androgen is there, they probably live longer. If the androgens are not present, then they do not live as long. Whereas the differentiated cells, they are androgen dependent, meaning that when there is no production of androgen, okay, then the cells usually start dying. Okay, so we call it as androgen dependent. I would also like you to remember that different tissues have different needs in terms of signals coming from the rest of the body. Okay, in this case, the prostate gland is dependent on the signal coming from androgen. The signal is androgen, right? So when there is, it is kind of signal the body sends. If the androgens are not produced in sufficient quantities, maybe these cells do not live as long, and the tissue kind of probably becomes smaller. But when we have excess of them, then they can probably divide and live longer. So if you look at the cross section of the prostatic gland, as we already discussed, we have a basal layer, right? These are the basal cells here. And The lamina is a kind of extracellular matrix-like thing that is present as the outer covering of the gland. And then we have the transit amplifying cells, TACs. These are called, you can also call them as intermediate cells. And then we have some cells, the luminal cells, of course. These are the luminal cells, and then they secrete. Here are the luminal cells, yes. And they secretions into the lumen. What we are looking at here are, you have so many different types of cells here, right? We have basal cells, luminal cells, neuroendocrine cells, and intermediate cells. And each of them seem to have their own or unique cell surface markers. In basal cells, they are CK5 plus. You don't need to mug up, but it's worth trying to at least acknowledge the types of cell surface markers they have. The basal cells also has CD44, right? And other things as well. The luminal cells, if you look, they have a different type of cell surface markers, CK8 plus, CK18 plus, and so on. Androgen AR plus would be probably androgen receptor, and the PSA, prostate specific antigen, PSA. And there are also several, the transit amplifying cells may have a different other cell surface markers. What we are talking here is general, the normal tissues and the cells of a prostate gland. I just wanted to put in about a word about neuroendocrine cells that are present. These are specialized cells. They produce neuropeptides, which are recognized, which receive signals from other sources, and they influence these cells here, and because of which, when to produce, the prostatic cell secretory functions are modulated by these neuroendocrine cells. Okay. So there are two different signals, right? One of it is the androgen, right, which is important for viability of the luminal cells or other differentiated cells. And the other one is how the prostate gland is integrated with the rest of the body is through neuroendocrine cells, because they bring in the signals from outside, when to secrete and when not to, and so on. Excuse me. Sorry. So let's have the similar one, just to have. This is prostate stem cell, which will divide into transit amplifying cells, and transit amplifying cells will further divide into basal cells, neuroendocrine cells maybe, and luminal cells, right? These are a total of, you can assume there are to be, uh, prostate stem cell is one cell type, and TACs are actually intermediate cells, so they're not, they will give rise to some other cell types. And then we have basal cells, which are the outer lining cells, which are attached to the basal lamina. We have neuroendocrine cells, which they put a question mark, which means the origins of them are not really are clear. Okay, there is a suspicion maybe they're arising from the prostate stem cells. And then we have a vast abundant and functional cells, that is are the luminal cells. And as I described, the luminal cells are androgen dependent. That means their survival or longevity is dependent on androgens. We will see the regulatory pathway as well as we progress. So we have, we will go to the slide, and that will help us better. Here is what is important about um, the androgen, why and how androgen can influence the luminal cells of the prostate gland. So testosterone, that is produced in the male sex hormone. It is a steroid, so it can pass through the cell membrane easily. Just give me a minute. It can pass through the cell membrane, and once it is inside the cell, it can be acted upon by 5 alpha reductase. Reductase means by adding 2 hydrogens, it makes dihydrotestosterone. Testosterone is acted upon by 5 alpha reductase and makes it as dihydrotestosterone. And now the dihydrotestosterone is active and it can bind to AR, or that is the androgen receptor. So usually the receptors are on cell surface, but this is a cytoplasmic receptor. Because if you remember the ligands that cannot pass through the cell membrane, ligand that cannot say peptides or proteins, they usually have cell surface marker. This is a cell surface marker, right? Surface marker. Whereas ligands like the steroids and others that can pass through the cell membrane, they are present. They have their receptors for them are usually in the cytoplasm. You call them as cytoplasmic receptors. Here, androgen receptor AR is a cytoplasmic receptor. And once the dihydrotestosterone is bound with androgen receptor, they form dimerization. That means 2 of the androgen receptors bound with the dihydrotestosterone will bind together like this. Dimerization happens, and also phosphorylation happens. They gain a phosphate group like this. And once something like this has formed, a complex like this, with the post-translational modification of phosphorylation, it will translocate into the nucleus. All this is happening in the cytoplasm. And now it will get into the nucleus. In the nucleus, this whole complex will act as a transcriptional factor. They will bind to ARE, or androgen response elements, right? And that is where the androgen receptor binds along, if only it is bound with dihydrotestosterone and these modifications of phosphorylations are there, they bind there, and then they will activate the transcription of several genes, those that include survival, growth, and prostate specific antigen production. And that is why this especially happens in the luminal cells. If we go back in this case, um, in the luminal cells, if you see, we said AR plus, that is the androgen receptor plus. Okay. So if there is no testosterone, okay, if we did not have testosterone here, then there is no dihydrotestosterone. In that case, the androgen receptor will be bound by a protein that is heat shock protein, and then it is not active, and so there will not be any activation of these genes. So the growth will not happen, and survival will be compromised, and cell might undergo death. Okay, that is why the luminal cells, especially, they are highly prone to, or they are dependent on these androgens for survival and growth. So this is what we have learned until now is general biology. And what may happen occasionally is that the prostate gland, especially as the men reach ages around 60 or so, and if there is genetic predisposition and any other factors, some of the cells may become androgen independent, or they'll start undergoing dividing rapidly, and that is what will eventually turn into a cancer. And then it will reach the blood, and then stage 1, stage 2, stage 3, and stage 4, it will move to the different other organs and then cause secondary tumors, and that is eventually the person will die. Okay, how to treat in this sense case? Before that, we will get into some of the features and some of the symptoms. A normal prostate gland is something like this. This is the bladder, and here we have the prostate gland. And as I was describing about cancer, some of the things that we need is space. They need nutrition, right? That is when they take away the majority of the nutrition of the body is becoming weaker. And when they take away space of other tissues, then the function of other tissues is usually also compromised. So in this case, bladder has enough space, right? That is where urine is supposed to be collected. And because there is space and the other parts are occupied, so this is a good place for the tumor to grow into because the space is available. And when the space is taken up, you have less space for the amount of urine to be collected. So as soon as that means this will get filled up very fast because it is less in space, and so the person has to go for urination often. So that is the reason why we have frequent and urgent urination, and the person cannot put pressure on his on the belly, so the urine falls usually, because this is also painful when we put in pressure. This may cause pain, and you always have the feeling that the bladder is not empty because the space is already occupied, and it gives a sensation that the urine is still not the bladder is not empty. There can be hematuria or hemospermia, meaning that mixture of or bleeding along during urination or during ejaculation or along with sperm volume, and might have reduced, and impotence, that is viable sperm may not be there. And in general there is a lot of discomfort, and it's like something is sitting in the body. And all this is because this tumor has occupied the space and then in the bladder, and usually it is growing, which is what will cause the problems. While we learn about many of these diseases, so one should not be alarmed by it. The more we learn, the more we become also sensitive about it, including me in general. So we should not suspect the worst immediately. Okay, as long as if the same things happen after the age of 60 or so, then probably we might have to take care as a child, and then we grow into the adults, and after adulthood we slowly degrade or degenerate in our capacities and in our metabolism activity and many things. About 60 or so approximately, I'm on the optimistic 60 side. After 60, probably the problem should be taken care of. I mean, one can be suspicious about something is wrong. Until then, probably it is not as much. The probability is very less. But there are rare cases. So we don't have. The point is that we don't have to be alarmed about something is happening and do I have it. Googling and other things will create a negative impact on our psychology, and then our body can really get sick if the mind is sick. So the types of prostate cancer are, as you say, we don't have to mug up: benign cancer, prostate hyperplasia, prostatic intraepithelial neoplasia, and invasive adenocarcinoma. This is the most detrimental of all these things. This is all right. It's not as malignant. This is early stage. You can consider it in stage 2, stage 3, and this one is kind of stage 4. Okay, so if we know about prostate gland, I mean, we know about prostate gland, and we are discussing about prostate cancer, and how can we isolate or find out prostate cancer stem cells, right? And also the question to start with, as a general normal tissue, which of these cells, we have discussed about 4 different cells: basal cells, luminal cells, neuroendocrine cells we have discussed. Where are the prostate stem cells? right? That is what we want to know. Or which of these cells are prostate stem cells. So apparently, these stem cells are present in the basal layer. Some of the cells of them are the prostate stem cells, which give rise to intermediate or transit amplifying cells, and they give rise to the rest of the cells. Okay. The idea or the question here is, what would be the cell surface markers of the prostate stem cell? So I hope you remember how to identify or characterize the cancer stem cells. If it is, uh, and here, if it is prostate cancer stem cells, say for example, then how would you do it? You would have taken the tumor, make a single cell suspension using FACS cell sorter by using antibodies against different things that we have discussed, maybe CD44 and several other types, and then test them in SCID mice to find if they have the ability for three things, right? First thing is tumorigenicity. Second thing, self-renewal. And the third is differentiation, heterogeneity, right? So what has been found is what we are trying to discuss here. The as I said before, the basal stem cells, basal cell layer, that is of the prostate cancer, prostate gland, and here we have luminal cells like this. On top of it, we have basal lamina, which is kind of extracellular matrix. And I discussed that some cells within the basal layer happens to be the prostate stem cells. And how do we identify them is the question, right? As normal things. So all the basal cells have CD44 plus. And then they did FACS again. Through FACS, they have separated all the basal cells. Next round of FACS, what they did was, again, it is a FACS. For the next round, they used antibodies against integrin alpha 2 beta 1. If some cells within the basal cells, some of them have integrins in high concentration on the cell surface, high numbers, and some cells had very less. So using antibodies against the integrins, they can still separate. Some of the cells will give very intense color or fluorescence or signal, and the other cells will give lesser, right? So they separated them based on whether alpha, the integrin, is high or low. And then they've taken CD44 plus, integrin alpha 2 beta 1 high, and performed again another FACS. And this time they used antibodies against anti-CD133. So you'll get two populations, that is CD133 plus and CD133 minus. CD133 minus cells happens to be the transit amplifying cells, whereas the CD133 plus cells happen to be the stem cell population. So if you look at it, how did they do that? They used in vitro proliferative potential. How long can a cell divide? How many times can they divide? So they would have taken basal stem cells, FACS, they would have taken these two different cells in different tubes and performed, checked which one are growing for a longer duration, that is dividing. Proliferative potential means dividing, how many times they can divide. They found that this is dividing more. They want CD44 plus and integrin alpha 2 beta 1 high. Then again, they separated these cells and made CD44 133 plus and 133 minus, and they tested which ones can divide for a longer duration. And they found that CD44, this is the potential, you can see this given as a kind of equation. CD44 plus, integrin high, and CD133 plus has the highest proliferative potential. And therefore, They are the stem cells, prostate stem cells. Okay. So we will also discuss about what is meant by proliferative potential and proliferative rate in some of the slide. Maybe it is here. Yes. So these are this is I actually am more interested in this particular one, the graph that is shown here. On one side, they're showing proliferative potential, and this you can think on time. Okay. There are two lines indicated. Which one is proliferative capacity and which one is proliferation rate? Let's understand these terms first. Proliferation rate means proliferation by time. In a given time, how many times can cell divide? Cell divisions per time. Okay, per unit time. Proliferation capacity is something. How many total cell divisions? Total cell divisions can it undertake? There is no time limit. Can you undergo 100 cell divisions, or is it only 50 cell divisions? So some cells have very high proliferation rate and some cells have very high proliferative capacity. Capacity is total capacity, total number of cell divisions that a cell can undergo. So if you look at the black line here, the number of proliferative potential, or how many times cell division is happening, it is happening dramatically. So if you look at this is indicative of proliferative rate. Okay, when the cells will divide, say for example the transit amplifying cells. Okay, they divide much more rapidly. That means they have high proliferate uh, capacity. Transit amplifying cells, they have high proliferation rate. Stem cell, the one that is present, PSA or any other general stem cells, their proliferation rate is low. Rate is low. While their proliferative capacity, that is stemness here, right? Continuous ability to divide continuously, stemness is very high. As the cell is dividing into TAC, for example, okay, proliferative capacity will become lesser. Proliferative capacity will become less and proliferation rate will be high. And eventually it will form, say, luminal cell. In luminal cell, ability to divide, proliferative capacity is minimal and proliferation rate is also minimal. I hope you understood this point. I'll try to repeat again. Stem cells, adult stem cells, proliferative capacity or the stemness is very high, but the rate at which they divide is very low. They are majority of the times dormant or quiescent. They will only divide when there is a need. But transit amplifying cells, their proliferation rate is very high. That means as soon as they form, they will divide much more rapidly. I mean, not for too many, not for many rounds, because they have reduced capacity. Capacity is reduced. So maybe they will go 3 rounds or so. And as they are dividing, the proliferation capacity has come to zero and their proliferation rate also comes to zero. This is what happens to the completely differentiated cells. Okay, this is a good way to understand the general organization of tissue as well. Okay, so we will just. So, what is the one of the uh, ways of how to treat cancer stem cell? Because what we have learned is androgen is important for luminal cells. right, for the survival and growth of luminal cells. And if we remove the androgen, then the luminal cells may die as well. That's a good logical way to continue to prevent the or control the prostate cancer. So in the some days they would have performed castration. Castration is removal of testis. So if the testis is not present, testosterone may not be present, and because of which the mature cells, they will die at a faster rate. That is in normal ones. But in malignant ones, they become, even though the androgen is depleted, some mutations, because a cancer, once it is formed, it's like a mill producing more and more mutants, and some of them may gain functions which can grow in any kind of situation the body may be present at that time. So there is a random chance always. So some mutations may come about, in which case the cell can grow even though the androgen is absent. Okay, we will see in the slides to come how that is possible. Let's see this again. Let's review what we have discussed. So from testosterone and adrenal gland, we get these androgens. You can think of testosterone and androgen, which will be converted by 5-alpha reductase to dihydrotestosterone, and dihydrotestosterone will bind to androgen receptor and dimerization, phosphorylation, and that is translocated into the nucleus and will allow the transcription of several genes that are involved in growth and survival. We are talking about, say, androgen, testosterone. If castration, that is removal of testis, happen, there is no testosterone production. So there is a possibility that the prostate gland, the luminal cells, at least they would die because they're dependent on their survival and growth on the testosterone. So what may happen, this we already have seen, and this is the general canonical way of how the androgen receptor, the cell signaling pathway, works here. So before we get in, what are the therapies that are available? One way of controlling is by androgen ablation, by removal castration, for example, or other medicines that might interfere in the availability of testosterone to enter into the cell. We can also probably use some inhibitors against alpha reductase as well, because dihydrotestosterone is the active form which will bind to the androgen receptor. If we prevent the formation of dihydrotestosterone, then too we can prevent the survival and growth of the luminal cells. The normal, what are the classical ones available are this prostatectomy, that is, they would remove the prostate gland. If it is detected early in the lifetime, early stage, then we can probably remove the prostate gland. As we discussed, prostate gland is non-essential, so the person may not have big issues except for some urinary inconsistency and impotence. As I said, the prostatic fluid is kind of important for the sperm survival or longevity of the sperm. And so storage or shelf life in the seminal vesicles could be very less, right? But otherwise, the person can lead a very normal life. And what we should understand is this usually happens after the reproductive age, if there is prostate gland. So reproductive age starts, say, for example, from 15 years onwards. And by the time someone is 35 or 40 years, they would have had kids. So even if prostate gland was removed and there is impotence with regard to that, there are no big issues, right? In social living, I'm talking about. Radiotherapy, this is probably a curative one in the earliest stage if we can do that. But there will be so many problems, impotence, along with, because radiotherapy is, we are directly putting radiation on the area near the where the prostate gland is, that is very close to the bladder, the rectum, and many other things. So there could be, during the time of treatment, there could be a lot of problems with that. And hormonal agents, they can be used, as it discussed, that is to reduce the amount of testosterone produced, or to bind testosterone or eliminate it from the body. You can have many of those things that can be done, but then there could be a lot of function problems with reduced libido, impotence, and other things. Chemotherapy is against, there are some names here, docetaxel, and so on. And there too, of course, chemotherapy has its own problems with, because chemotherapy is generally given as a systemic medicine. So while it might do a function at one place, it might interfere with the general body somewhere else. Something like neurotoxicity, for example, or anemia, which might influence these things, will be, these are some of the side effects. Okay, I'll just talk about this one, and I will stop. Androgen independent survival. In the previous time, we were discussing about androgen ablation, where we are somehow suppressing or eliminating the production of testosterone. But the problem is that some new androgen independent cells can come up. That is, of course, because of the mutations. Right? As I said, cancer, once it has developed and it is rapidly dividing, and we also discussed that in the introductory lesson on cancer, mutations are kind of proportional to rate of replication. Right? In cancers, rate of replication is very high. That means the probability or the mutations also are high in cancers. So if we try to treat it in one way, because mutations are continuously happening, some of them may be resistant mutations, resistant to the therapy that we are using. And then the cancer is kind of adapting to whatever we are trying to treat. So how does this happen? Let's just recollect. We have testosterone, which will enter into the cell. It will be converted into dihydrotestosterone. It will bind to androgen receptor, and the androgen receptor will go into the nucleus, and and there you will have growth genes expressed. Genes that are involved in growth and survival are expressed. Right? So a mutation can happen mostly it can happen in the androgen receptor. What are we saying? Androgen receptor gets activated and moves into the nucleus only if it binds to dihydrotestosterone normally. What if there is a mutation because of which androgen receptor dimerizes and phosphorylates and becomes active all the time? So here are some of the scenarios. This is the normal one. Testosterone and dihydrotestosterone, androgen receptor, and then inside, and then growth and survival. Where in some cases, where there is hypersensitive, in the sense if you have excess amount of androgen receptors, that means we will have excess activation of the genes here, and that might result in excessive growth and survival. Right? This is having higher number of androgen receptors is the problem here. Everything else is normal. And how can you have higher number of androgen receptors? By directly or some indirectly. If some mutation happens that is influencing the expression of androgen receptor, then it will become hypersensitive. The other one is called as promiscuous, in the sense that normally the androgen receptor is the ligand for it is dihydrotestosterone, but because of a mutation, the active site may be change to something else. And now here a different molecule can bind, and if it binds and that molecule is abundant, then androgen receptor gets activated, phosphorylated, dimerized, and then it can act as lot of activity. We call it as promiscuous, meaning that the choice has changed from one to the other person. It is actually, it should have been for dihydrotestosterone, but now it is some other molecule similar to that of dihydrotestosterone. The specificity has changed, or the liking or affinity for a different molecule has increased. There is another way, that is by activation of androgen receptors by other signaling pathways. Somehow, because of any mutation, if these androgen receptors become activated or connected or crosstalk between, say, other signaling pathways, then too we might end up with having prostate cancer. These are androgen promiscuous and outlaw. These are androgen independent growth, right? Even if testosterone is not present, okay, we were discussing about it. If we remove the testosterone, then these androgen receptors will not activate the cell. Now, in the cases, any mutations that make the androgen receptor like this, responsive to other molecules or be influenced by other proteins, then even if there is no androgen, if there is no testosterone, it doesn't matter. They will still, the androgen receptor is active and will cause continuous growth of the cell and survival. Okay, I will stop here, and what I will just conclude here is these are some of the mutations people have identified, which are reasons for promiscuity. This is wild type androgen receptor. It should be binding to dihydrotestosterone, right? And because of some mutation, they might now be binding to some other compounds. Okay. Here is another mutant. These are some of the agonists, that is the ones that are binding. Antagonists are those that don't allow the binding, right? So here are some of the mutations, and I will discuss in the next class. I think time is up. Okay. If you have any questions, please, you can ask. Yes? No questions?
No questions.
Okay. Yes, feel free to ask by message if you want. Okay. Thank you.
Sir, regarding
Yes, please.
PAAC, so the syllabus, sir,
Syllabus, when is the exam, did we say? We
October
said the
30th. Sir.
exam is on October 30th?
Yes, sir.
Yes. So whatever we finish until the coming Monday.
Okay.
Oh, Monday we have a class? Yeah, until Tuesday, 26th. Whatever portion is finished until 26th, that will be part of syllabus for
Okay.
CAA. Okay?
Okay.
Yes. Thank you very much.
Thank you, sir.
Thank you, sir.
Yes, see you. Bye.
Thank you, sir.